Hey guys, welcome back. Today we're going to be going over chapter 14 from Campbell Biology in focus, and we're going to look at how genes turn into proteins, or better yet, how they code for proteins. So the overview is going to be the flow of genetic information. So this is the central dogma of biology that we'll talk about, but basically your DNA is the blueprint, and then your DNA is turned into RNA, which can then be translated into proteins. So the overview here. So the important content of genes is in the form of specific sequences of nucleotides in DNA. Because previously when we talked about DNA, we said that it's really the order of the nucleic acids in each individual that makes your genetic makeup unique and specific to you. So the DNA inherited by an organism leads to specific traits by dictating the synthesis of particular proteins. So these proteins are the links between the genotype and the phenotype. So your actual genetics, the alleles, what you have, what you've inherited, and the phenotype, what you physically look like. Our gene expression, it's the process by which DNA directs protein synthesis, and it includes two distinct stages, transcription and translation. So these are the two steps of our process of protein synthesis. So transcription, the C comes before translation, the L, so they are in alphabetical order to try to keep that straight in your head. Okay, so we're going to look at different studies first. This is the most famous one to kind of connect the whole gene to protein connection here. So in 1902, a British physician, Archibald Garrod, first suggested that genes dictate phenotypes, so the way that something looks, through enzymes that catalyze specific chemical reactions. So he thought that the symptoms of inherited disease reflect an inability to synthesize specific enzymes. So cells synthesize and degrade molecules in a series of steps, which is called their metabolic pathway. So then we're going to look at some of the studies here for Beadle and Tatum. So what they did is they actually disabled genes in bread mold. So like, when you leave bread out, it gets kind of funky looking. Great. That's what they were studying. So they looked one by one at different traits to see what actually affected the phenotype. What caused a phenotypic change? So what caused a physical change? What were they messing with that caused a phenotypical change in this bread mold? So they chose this because it grows very quickly. It grows through budding, and it's also a haploid bread mold. So it's easier to detect recessive mutations because you're only dealing with half of the genome there compared to a normal organism that's a diploid organism. So they studied mutations that altered the ability of the fungus to grow on minimal media. So this is essentially a minimal media. It's just like the minimum requirements for life. So you have a petri dish, you're able to grow different colonies of the fungus on the minimal media because the media that's actually in the petri dish contains everything that particular organism needs to grow, just the minimum requirements. So they were seeing what do they have to turn on, turn off in order to affect how it grows on this minimal media that every one of these fungi should be able to grow on. So here's their experiment. So they were working with a neurospora cells, which are just a type of fungus. So the individual neurospora cells were placed on complete growth medium, and then they were subjected to x-rays. So we know that x-rays cause mutations. Okay, so they were seeing then what happened to these colonies. So each of the surviving cells forms a colony of genetically identical cells, which makes them, again, really nice to study because they are all identical. So they took those cells and they put them on the minimal media, which is what we're seeing, like, can they grow on minimal media? And you'll see here that there was no growth. So something happened because initially these cells were able to grow on minimal media. So after you expose them to x-rays, now they can no longer grow there. So something had to change about their metabolic pathway to make them now completely unable to grow on this minimal media. Okay, and then the control, of course, was using the wild type that you did not hit with a whole bunch of x-rays. They still grow on the minimal media. So this is exactly what you would expect. Now you absolutely have no growth. So what they discovered is that they put these cells that have been subjected to the x-rays that don't grow on minimal media, they put them on minimal media that had one more ingredient. So they added amino acids, which we know is like a building block of all of our proteins. They added different amino acids to different petri dishes, so minimal media plus one amino acid until they got growth. So that's what they did here. So the mutants were placed on a series of vials, each containing minimal media plus one additional ingredient. And it was different for each one of those series of vials, right? So then they saw growth, and what they actually saw was growth on arginine. So minimal medium plus arginine showed growth. So this tells you that something happened when the X-rays hit these cells that affected their ability to create or utilize arginine. So it had to be now present in the growth medium for them to be able to grow. So that's how their whole experiment works. So let's talk about that a little bit more. So the researchers amassed a value collection of our Neurospora mutant strains and cataloged them by all of their different defects that were created by, turning on and off certain genes. So for example, one set of mutants all required arginine for growth. And this is one of the things that they found out after subjecting them to X-rays. So it was determined that from different classes of these mutants were blocked a different step of their metabolic pathway for arginine biosynthesis. So when you actually affect particular genes, it's now affecting their overall growth, which you could visibly see because they're trying to grow them on all these different types of media, right? So that's how they were able to see that it was the arginine pathway that was actually disrupted by these defects that they were introducing to the fungus. Okay, so we're going to go through the products of gene expression. So some proteins are not enzymes. We know that like in freshman biology, like that's what they teach you, like all enzymes are proteins. Well, proteins have a whole bunch of different jobs, and enzyme is just one of those jobs. So some proteins are not enzymes. So researchers later revised that one gene equals one enzyme to be one gene equals one protein, because not all proteins are enzymes. So this is now like a better definition of one gene creates one protein. And that's a pretty good definition. So many proteins are composed of several polypeptides. Many peptides, peptide bonds exist between amino acids to hold, the amino acids together to create a protein. Each of which has its own gene. So each particular gene codes for a particular protein. That's how that works. So Beadle and Tatum's hypothesis is now restated that one gene is one polypeptide, which makes sense because a polypeptide typically makes a protein, but remember that you can have several polypeptides required to make certain proteins. Okay, it is common to refer to gene products as proteins rather than polypeptides. That's just kind of like common convention. So our basic principles of transcription translation, those remember I told you are the two steps for protein synthesis. And all of this is about how do we get a protein or a polypeptide, whichever, you can kind of use those interchangeably. So RNA is actually the bridge between DNA and protein. So this is the central dogma of biology that I brought up at the beginning. So like all the overarching themes of biology is your DNA is the blueprint of life, which is then copied onto RNA, which kind of acts as this bridge to help us create our proteins in protein synthesis. Again, how to make a protein. Synthesis means to make, and a protein is obviously a protein. So RNA is chemically similar to DNA, but RNA has a few differences. So first of all, the R in RNA is a sugar called ribose. And then, of course, the base uracil is in place of thymine. So instead of A to T, A is going to now pair with U. Okay, and that's only in RNA. Your DNA is still chilling, A and T, apples grow on trees. That's still a thing in DNA. We're talking about RNA. RNA, instead of using thymine, uses uracil. So RNA is usually single-stranded, so that's another big difference from our DNA molecules. Getting from DNA to the protein requires two different stages, of course, like I said, transcription, translation. They occur in alphabetical order, so don't forget that on exams. They occur in alphabetical order. The C comes before the L. Okay, so transcription, this comes first, is a synthesis of RNA using the information embedded on our DNA. I like to think about it like that you have your cell, and that you have a nucleus, and your DNA is inside of that nucleus, and it's stuck there and it can't ever leave. But your ribosomes can be, attached to our organelles out here. They can be free floating in the cytoplasm. They can be made a little closer to the edge of the cell membrane. Right, but we have our DNA that's stuck here, and we have our ribosomes all over the place in the rest of our cell. How is that information going to pass from the DNA to the ribosomes? The ribosomes are not inside that nucleus, so how can they communicate? So we use something called RNA to do that. So RNA, like I said, is the bridge. So transcription is step number 1 in protein synthesis, and it's how we make RNA. So transcription equals DNA to RNA. That is transcription, DNA to RNA. Okay, so we're actually going to create something called messenger RNA, called mRNA. So transcription specifically produces mRNA. So this is like a little bit more specific there, mRNA. And then translation, step 2, is a synthesis of our polypeptide, which is going to use the information that mRNA copied from our DNA. Because remember, DNA is stuck inside the nucleus. That's not where the ribosomes are. So we have to have it's like playing a game of telephone, you have to pass along the information. That's exactly what's happening here. You're making a copy of the information and passing it on to the correct organelle. In this case, it's going to be a ribosome. So ribosomes are the site of translation, which again is step number 2. So transcription, DNA, we're going to copy the code onto mRNA. And this, of course, happens inside of the nucleus because that's where the DNA is housed, right? And then step number 2, translation occurs at a ribosome. And we're going to take that mRNA, we're going to add a few other guys, but we're going to turn that into a polypeptide, also called a protein. So those are your 2 steps. That's the overall thing for protein synthesis here. Okay, so in prokaryotes, translation of mRNA can begin before transcription is actually finished. So they're happening at the same time. Because think about, they're a lot, their chromosomes a lot smaller, they're a lot simpler in structure and function. So in eukaryotes, the nuclear envelope separates transcription from translation because the nuclear envelope, remember, that's the DNA is inside of the nucleus. So that's like a barrier before we have our little mRNA that can be passed through that nuclear envelope to meet up with a ribosome. So remember that that's how that works. So it's 2 distinct processes in eukaryotes, whereas in prokaryotes, it's all happening kind of at the same time. So then in our eukaryotic RNA transcripts, they're modified through RNA processing to yield a finished mRNA because we have something called pre-mRNA, and then afterwards it becomes like big boy mRNA that's ready to go off and make a protein. Okay, so eukaryotic mRNA must be transported out of the nucleus to then be translated. Okay, so transported, so we made this in transcription and transport it out of the nucleus, as I drew up here for you, to then be translated again at the ribosome. Okay, so here's a picture of that happening. So in a bacterial cell, remember that I said that it's all happening simultaneously. You have transcription and translation that can be happening kind of all at the same time. Transcription does not need to be completely finished before you have translation start to occur. And remember that there is no nucleus here, right? There's no nucleus in this cell. So there's no physical barrier between the 2 of them. Whereas in our eukaryotic cells, we have a nuclear envelope that actually physically separates the 2 processes. So we have transcription, which remembers when we have DNA, and we're going to turn that into mRNA because we're going to copy the code onto mRNA. First, we have pre-mRNA, and then we're going to RNA process that to have our finished mRNA, which will then go out of our nucleus to meet up with a ribosome, which will be the site for translation. Again, translation, like I said, there's a few other players we're going to talk about, but you're using the code on mRNA to then make a protein. And again, this is protein synthesis. A protein is our final goal. Okay, so a primary transcript is the initial RNA transcript from any gene prior to processing. So when this is called our pre-mRNA. There's no processing, right? It's pre-mRNA. So the central dogma is the concept that cells are governed by a cellular chain of command. And of course, the commander of all of this is what? It's our DNA. And then we're going to turn that into RNA by copying the code. And then we're going to create a protein. And proteins then go and do all kinds of different functions in your body. So here's our central dogma. Central dogma of biology. This is it right here. So I'm just going to illustrate a little bit on here for you that DNA to RNA is a process that we call transcription. And of course, if we're dealing with DNA, it occurs in the nucleus. And then on the other side, RNA to protein, this is a process called translation. And this, of course, occurs at a ribosome. And that can be a free or a bound ribosome, just depending on which particular protein is being created. Okay, the genetic code. So these are some questions that we have here. So how are the instructions for assembling amino acids into proteins encoded into DNA? How does that work? There are 20 different amino acids, but there are only 4 nucleotide bases in DNA. A, T, G, and C. So how do we get from 4 to 20? That's a good question. How many nucleotides correspond to an amino acid? Another great question, because again, we have 4 nucleotides in DNA, and now suddenly we have 20 different options. So we're going to look at how all of this works together. Okay, so codons. Do you see this word codon? Okay, codon. It's the code for amino acids. Okay, it's a triplet of nucleotides. What does that mean? A triplet of nucleotides. 3, right? 3 nucleotides. That makes 1 codon. So the flow of information from a gene on our DNA to a protein is based on a triplet code. Now, this triplet code exists on mRNA. mRNA is just a whole bunch of codons put together. So the triplet code is a series of non-overlapping 3 nucleotide words. We're going to get into that in a minute. Okay, the words of a gene are transcribed into complementary non-overlapping 3 nucleotide words of mRNA. These words are then translated into a chain of amino acids. And amino acids, of course, are the subunit of a protein, so then they will form a protein. So the codons are on the mRNA. They exist in groups of 3, and that's what tells you which amino acids are needed to build that protein. So we're going to look at that a little closer in a second. Okay, you have your DNA template strand, and this is located inside the nucleus. We're talking about eukaryotes for the majority of this because we're selfish and that's what we are. Okay, transcription. You know now, because I've said it 150 billion times, DNA to mRNA. We are copying the code. Remember that mRNA is a single-stranded molecule, so it's going to look a little different. I also just told you that mRNA is made up of a bunch of what? Codons. And codons are what? A triplet of nucleotides. 1, 2, 3, that's a codon. 1, 2, 3, that's a codon. 1, 2, 3, look, another codon. And finally, a last codon. That looks like a 2, that's a 3. So here we have 4 codons on this mRNA molecule. Codons do not exist anywhere else except for on our mRNA. I'm going to repeat this because later when we're having to use charts and things, you're going to start messing it up. So codons exist on mRNA. Codons are a triplet code of 3 nucleotides. Uracil, guanine, guanine. Uracil, uracil, uracil. Guanine, guanine, cytosine. This is the code. Okay. Then translation, which we know takes that mRNA copied code from DNA and turns it into a protein. Remember, I said codons code for what? Codons code for amino acids, and amino acids are the subunits of a protein. A whole bunch of amino acids put together with polypeptide bonds equals a protein. Okay, so UGG, codon number 1, makes our first amino acid. Codon number 2 is going to make our next amino acid. These are just the first, typically the first 3 letters of the different amino acids. So like tryptophan, phenylalanine, glycine, and serine. It's up to you and your teacher whether you need to know those. My kids, you don't need to know all the names, but don't call it PHE. Okay, either just write it or understand that it represents phenylalanine. Cool. And then of course between these are polypeptide bonds because that's what actually holds a protein together. That's why a protein is also called a polypeptide. It's the same thing. Okay, so that's the overall picture of what we're working with. So during transcription, one of the 2 DNA strands, because remember DNA is double-stranded, DNA, double-stranded, the D's, double-stranded DNA. Okay, cool, you're with me. Okay, that's called the template strand. It's going to open up and you have a template strand. Now, we talked about DNA replication. We said if you know half the information, you can figure out the rest of it because A always has to pair with T, G always has to pair with C. That's how it works. So this template strand is going to provide a template for the order of our complementary nucleotides, right? That's going to make our RNA transcript, which is going to be our mRNA. But of course, remember that it's using uracil. Okay, the template strand is always the same strand for any given gene. Cool. Okay, so during translation, the second step that happens out at the ribosome, the mRNA-based triplets called codons are read in the 5' to 3' direction, which we talked about previously. So each codon specifies an amino acid, one of 20 different amino acids, to be placed at the corresponding position along a polypeptide. So remember, our codon, you have all these different codons along your mRNA, and whatever letters they are, that triplet code tells you, hey, make this amino acid, whatever that amino acid is. I don't know off the top of my head. They all code for a specific amino acid. Then when you have all of your polypeptide bonds, you make a protein. So that's how that code works. The code on the codons, which is on your mRNA. Codon, code for amino acid. Cool. Okay, cracking the code. What is this code? It's a codon code for amino acids. So all 64 codons, there are 64 different combinations. You got your A, in this case U, because we're dealing with RNA, right? A U instead of T, G, and C. There's 64 different combinations because math tells you that there are 64 different combinations. Okay, that were deciphered in the mid-1960s. So of those 64 triplets, 61 of them actually make an amino acid, and 3 of them are stop signals, which means, hey, you've reached the end. You can stop now. They literally, what does it mean? It means stop. That's what it means. Okay. So the genetic code is something we call redundant because there are 61 codes for amino acids, but there's only 20 amino acids. So obviously, there's more than one code for the amino acids. Is that always true? No, but in a lot of cases, yes. And we're going to look at that specifically later. Okay, so the genetic code is redundant because more than one codon can specify a particular amino acid. So more than just 3 letters in a row, it could be a different group of 3 letters or a different group of 3 letters. Your codons can equal the same amino acid. That makes sense because there's 21 codes for amino acids, and there's only 20 amino acids. So you know that it's redundant, okay? But it's not ambiguous. No codon specifies more than one amino acid. So each codon equals an amino acid. So one codon equals one amino acid. And one amino acid, I lied to you. Oops. This is a question a lot of people like to ask. One codon is also a triplet, right? A triplet. So 3 what? Nucleotides. And what can we tell from this information? That if one codon is one amino acid and one codon is 3 nucleotides, that must mean that one amino acid is equal to 3 nucleotides. That's a question that a lot of people ask on exams. I have so many amino acids. How many nucleotides were needed to code for that? Well, if you have one amino acid, it's 3 nucleotides. Let's say you have 2 amino acids. 2 times 3 is 6 nucleotides, and so on. That's how that would work. Okay, so codons must be read in the correct reading frame, the correct groupings, in order to code for the specified polypeptide for that to be created correctly, right? So codons are read one at a time in a non-overlapping fashion. So you have your codons like that. That is a solid reading frame, okay? So if you shift that, then that creates mutations that we'll talk about in a minute, okay? This is a codon chart. There are two different codon charts. One looks like a circle. It's a little easier in my mind, but this one is another one that your professor may ask you how to read. Remember that there are 61 codes for amino acids and 3 codes for stops. Well, look at that. They already color-coded the stops for you. So the rest of them are 61 different codes for amino acids. So let's say that your first codon is CCA. Okay, my first base is a C. So I'm going to look over here, and here's my C. So I'm looking in this box right here. C. What's my second letter? It's also C. So second base up here, it says second base. You look at the C. So now we're looking in this column. Okay, cool. They overlap right here. What's my last base? A. Third base is over here. A is right here. And we're just going to follow that back right there. Here, CCA. That makes proline. P-R-O, proline. What do you notice about this? Did it really matter? Can I erase this whole mug like this? Boom. Beautiful. Okay. So we just had CCA, and that made proline. So CCA equals proline. What if it was CCC? What does that make? Proline. What if it was CCU? What if it was CCG? All of those make proline. That's the only option because that's what's in this box right here. Okay, proline. So that third position is called a wobble. I think it goes on to talk about this later, but it's called a wobble position because a lot of times, look at serine, this is the wobble position. It doesn't matter what that third base is in a lot of these situations. Right? That's the redundant code that we were talking about. Okay? Just so you understand how that works. Okay, the evolution of the genetic code. So the genetic code is nearly universal. This means that all organisms use A, T, G, and C in order to code their nucleic acids, their DNA. So shared by the simplest bacteria to the most complex animals. So genes can be transcribed and translated after being transplanted from one species to another. So what does that mean? I can take a gene that's just, it's just letters. This is my mRNA, we'll say. And I can, it's, it came from your DNA. It's probably a bad example for you. But I can put it into another organism. Probably should have used DNA, but hindsight, organism. Put that DNA, of course this is RNA, so again, sorry, into another organism. And because it's all the same pieces, ATGC or AUGC, if we're dealing with RNA, it can read it because all of the machinery code, it can read the same 4 pieces because that's what makes up everything. So that's how you get cool stuff like this. You got a tobacco plant that glows in the dark by it's expressing a firefly gene to glow. It is a plant that is glowing. You also have a pig expressing a jellyfish gene, again, glowing. You can also put different things in mice to make them glow. This is like genetic engineering. Genetic engineering. Taking genes from one organism and putting it into another organism. This is also how we get genetically modified organisms, also called GMOs. Now, everyone wants to lose their marbles and say, GMOs, oh my God, they're bad. They're going to give you cancer. Okay, let me ask you a question. Because now you are an educated individual because you just learned how genes go from one organism to another. And how is that? You cut out the DNA. You cut out the gene, which is DNA, that you want to put into another organism. Is DNA harmful? If DNA is harmful, would anything on the planet be alive? The answer is no. Right? It's not harmful. Would anything be alive without DNA? No, right? So genetically modified organisms or organisms where we took DNA from something and we put it into something else. DNA, A, T, G, and C in a certain order. That's it. It doesn't mean that there's chemicals in there that are going to poison you and give you cancer or 4 arms. That'd be kind of cool sometimes, but that's not how this works, right? You just have A, T, G, and C in a particular order that says, here's a gene. We're going to call it gene A. We're going to take gene A and we're going to put it into this thing over here to now make that thing over here express gene A. For instance, a lot of strawberry crops are genetically modified organisms. This means that they have DNA from another organism inside of them. A lot of times they've used flounder DNA, like the fish, like a flounder, like Ariel's sidekick, flounder, right? Because they can live in cold environments. Well, if you express that particular gene in a strawberry, does it taste like fish? No, it just means that they took that one gene that allows it to be in the cold and put it into strawberries. Therefore, strawberries can grow in the winter so we can have strawberries because they're delicious all the time. Okay? There's also like you can get certain, like elephants like grow to be really, really huge, right? So you can copy specific genes that allow them to get larger into produce and other foods in order to allow those organisms to get larger. Does it mean it's going to be like, kind of tastes like elephant? Is it gray? No, it's just the trait that allows them to surpass their normal growth that you're expressing in that. Also, corn crops. Corn crops are susceptible to all kinds of different, like locusts, crickets, things like that, eating them. Okay, corn's also delicious. So they took Bacillus thuringiensis, which is a bacteria, and they took a particular gene from that bacteria and expressed it in our corn, genetically modified organism. Therefore, the crickets and the grasshoppers don't want to eat it anymore because what was happening is all these locusts and grasshoppers, crickets, whatever, they're eating our corn crops and then, oops, now we don't have enough food. That's a problem. So just putting in this one little gene prevented these organisms from eating all of our food. Does that mean it's poisonous for you? Does that mean, tastes like crickets? No, of course it doesn't. Okay, it doesn't taste like crickets because they eat it. It's the Bacillus thuringiensis that it would taste like. But anyway, sorry, I'm a human. I make mistakes. Genetically modified organisms are not inherently dangerous for you. So anytime that someone tells you that they are, that is incorrect. Now, some people don't eat them for religious beliefs and everything. Go on about your bad self. That is for you. For me, I don't have religious beliefs that are going to prevent me from eating a genetically modified organism. If you want to make my strawberries bigger and able to grow in December and I can have strawberries year round, I'm all for it. Do it. Science is a beautiful, amazing, wonderful, magical thing, right? So that's what a genetically modified organism is. Now I'm going to step off my little soapbox. That's like a huge rant. I hate when people who are just like uneducated, ew, GMOs. You're saying ew to DNA. You have it. Are you saying ew to yourself? No? Okay. Do you like being alive? Great. It keeps you alive. You're welcome. Science. Anyway, sorry about that. We're going to get back into talking about proteins. So transcription is the DNA-directed synthesis of RNA. We're going to take a closer look at that. So transcription, DNA to mRNA is the first step of gene expression because if you're able to transcribe something, you'll go translate it and then express it. Okay? So let's look at this. So RNA synthesis is catalyzed by RNA polymerase, which pries the DNA strands apart and joins together the RNA nucleotides. So you've opened up the double helix of DNA, and now you are copying one of the template strands, right, onto your mRNA code. RNA polymerases assemble polynucleotides in the 5' to 3' direction. However, the RNA polymerases can start a chain without a primer. Here's what this looks like. Okay, so you have your RNA polymerase that's going to go through, and once we have our little gap that's opened up in our DNA because it is double-stranded, right? We have a little gap that's open. RNA transcription is going to start there. So we have an RNA transcript being created. It's going to copy from the template strand of the DNA. So, of course, if we're synthesizing in the 5' to 3' direction, that means that we have to be using this bottom one because remember, it has to be in opposite directions. That has to do with the backbone on the outside, the alternating phosphate sugar backbones. So that's what it's talking about. The 5' and the 3' is talking about the carbons, the directionality of the strands, right? So you have initiation, elongation, termination. My little numbers got messed up, whatever. Elongation, termination here. So termination is, of course, when you are at the end of your gene, then you stop transcribing. So now this is the complete RNA transcript. This is our pre-mRNA that'll go on to RNA processing before it actually contains exactly the code that we want to go and then make our protein. So DNA sequence where RNA polymerase attaches is called the promoter in bacteria. The sequence signaling at the end of the transcription is called a terminator. So you have the promoter, which is where it starts, and then you have the terminator, which is where it terminates or finishes. So the stretch of DNA that is transcribed is called the transcription unit. Okay, so like we call them genes, but like in bacteria, we have like transcription units. Okay, so there are 3 different stages of our RNA transcription here. So we have initiation, start, elongation, how it elongates, and then termination, the end. Beginning, middle, end, kind of. There we go. Okay, so RNA polymerase binding and initiation of transcription. So we have promoters that signal the transcriptional start point. Promoter promotes the start. Promoter promotes the start. Okay, and is usually extend several dozen nucleotide pairs upstream of the start point. So it's actually a little bit ahead of where you actually need to start your transcription. That's to make sure that you get the entire gene, and that's why we then process, because some of the stuff that you have might be like junk that you don't really want, and this is what you're after right here, and then everything that comes after that. That's why we edit, because the start points are usually a little bit higher up than where you're actually starting. It's upstream. It's before the gene. Okay, transcription factors are going to mediate the binding of RNA polymerase and the initiation of transcription. So the completed assembly of transcription factors in RNA polymerase 2 bound to a promoter is called the transcription initiation complex. So now we have all the pieces together that we need in order to start transcribing this particular region. So the promoter is called the Tata box, and it is crucial in forming the initiation complex in eukaryotes such as ourselves. So our promoters are called the Tata box. So we have transcription that we're dealing with right now, which of course is the opening up the DNA. We're going to create our pre-mRNA, and then we're going to go through and process that. So in eukaryotes, we have the Tata box. Okay, remember that this is the template strand for us because we're going to create our mRNA in the 5' to 3' direction. So we have to have it be the opposite because they are anti-parallel. So we'll be using the bottom, which is going to be here our Tata box. That is the promoter region. And then remember that I said that it's a little upstream from the start point. So this is actually where you want to start transcribing. You'll actually be transcribing kind of all of this, and that's why we edit later. Okay, and then you'll just go through until you'll have your transcription factors that are going to bind to the DNA, and that kind of helps stabilize everything. You have RNA polymerase 2 that's going to move in, and it's going to start laying down those RNA nucleotides to create our transcript there. That RNA transcript, once you go through elongation, will then be completed, and in termination will be stopped. Okay, so elongation. So now that we have our initiation, we are ready to go, right? We've got all of the main players here in our transcription initiation complex. So you're ready to elongate. So elongation. So as RNA polymerase moves along the DNA, it untwists the double helix 10 to 20 bases at a time. So transcription progresses at a rate of about 40 nucleotides per second in eukaryotes, which is pretty fast. Okay, so a gene can be transcribed simultaneously by several RNA polymerases. So this means that you can have a whole bunch of them that are doing transcription all at the same time at different places along the DNA, because DNA is huge. So you can have a whole bunch of different RNA polymerases that are in there making all kinds of different genes at the same time. Okay, so this is the overall picture here. You've got your direction of transcription happening here, because remember that we're using a template strand that is a 3' to 5', which means that we are synthesizing in the 5' to 3' direction. And we're just going to be adding in our little RNA nucleotides through elongation. Termination. When you're done elongating and you've reached the end of the gene that you're trying to create the protein for. So this the mechanisms of termination are different in bacteria and eukaryotes. So in bacteria, the polymerase stops transcription at the end of the terminator, and the mRNA can be translated without further modification. So you actually don't have to go through mRNA processing in prokaryotes. You have to do that in eukaryotes, though. So in eukaryotes, the RNA polymerase 2 transcribes the polyadenylation signal sequence. the RNA transcript is released 10 to 35 nucleotides past the poly-A polyadenylation sequence. And we're going to talk about that in a second. So eukaryotic cells modify RNA after transcription. This is mRNA processing or RNA processing. So enzymes in the eukaryotic nucleus modify pre-mRNA because it's got other crap in there that we don't want, right? Has other things in there that we do not want. So we just want to get our finalized mRNA to then go on and create our protein in translation. So before the genetic messages are dispatched to the cytoplasm, they have to be processed. So during RNA processing, both ends of the primary transcript are going to be altered. And this is for protection, but also for labeling purposes to help the ribosome attach. So usually some interior parts of the molecule are also cut out, and other parts are then spliced back together. If you remove something, you can't just have two pieces willy-nilly running around. You got to put them back together. So this is splicing that we're going to be talking about too. Okay, so the alterations that we're going to make here. So each end of the pre-mRNA molecule are modified with a particular way. So the 5' end receives a modified G nucleotide 5' cap. So we just call it 5' end gets a 5' cap. The 3' end gets a poly-A tail. That's that polyadenylation that I was talking about earlier. Poly-A, polyadenylation. So these modifications share several different functions. So they help to facilitate the export of the mRNA into the cytoplasm, because remember, it's in the nucleus and it's got to get out into the cytoplasm. It's going to protect the mRNA from hydrolytic enzymes that are present in the cytoplasm, and it's going to help the ribosome attach to the 5' end. So like I said, it helps to initiate translation, and then it helps the mRNA, make its journey from point A to point B, which would be the nucleus to the ribosome. Okay, so now we are in RNA processing. So we are taking out the pieces that we don't want in our pre-mRNA. We are adding our caps on and our tails, and then we are going to send that finalized mRNA out to translation. So here we have our 5' cap, which will help the ribosome identify, hey, this is mRNA. Hey, it contains information for a protein. Let's read it. Okay, and then our poly-A tail is going to help from preventing degradation of this while it's kind of moving around the cytoplasm. Okay, so split genes and RNA splicing. So we're going to look at how you cut pieces out, and it's kind of counterintuitive, but I'll explain that in a second. So most eukaryotic mRNAs have long non-coding stretches of nucleotides that lie between the coding regions. So this is considered to be like, quote, like junk. Like we don't really know what it's there for, but we know that if we take it out, it's like detrimental like to the organism. So we just edit our mRNA. So mRNA contains different regions called introns and exons. So the non-coding regions are called introns. Again, non-coding are called introns. The other regions that we want are called exons, and they're usually translated into our amino acids. So I'm just going to make this simple. Bad, good. Because exons code for amino acids. That's what we want because we're trying to make a protein. The introns don't code for anything. They're intervening. That's why they're called introns. To me, it sounds like they stay in and exon exit, but it's the opposite of that. They try to play us with these words here. So they're intervening. So they're interrupting. They need to get out. So these get cut out and these stay in. So that's a little confusing, but just think about it. Introns interfere, gotta go. Okay. RNA splicing removes the introns and joins the exons back together because you can't have like a spliced mRNA. It's got to be put back together, creating the mRNA molecule that is continuous with the coding sequence. So this is what that would look like. So you have introns. Remember, they interfere. They have to be cut out. Our exons are what we need to stitch back together. So then this is our finalized mRNA, and it will go into the process of translation. Okay, so many genes can give rise to 2 or more different polypeptides, depending on which sequence, which segments are used as exons. So this process is called alternative RNA splicing. RNA splicing is carried out by a spliceosome. It's very aptly named here. So spliceosomes consist of proteins and small RNAs. So here's kind of what this looks like. So you have a spliceosome. It's got some proteins. It has small RNAs that are going to go through and help edit the introns out, and then the exons will be put back together. So the introns interfere, they are removed, and then our spliceosome is going to help us put together our exons to create our continuous code to make our protein. Ribozymes are RNA molecules that function as enzymes. So that makes sense, like ribo for sugar, that's in RNA, and then enzymes, so ribozymes. So RNA splicing can occur without proteins or even additional RNA molecules. The introns can catalyze their own splicing in some cases as well. So translation is the RNA-directed synthesis of a polypeptide, and we're going to take a closer look at that. So now this is translation. So transcription, the whole point was to get a finalized mRNA molecule. And once you have transcribed it, you've capped it on one end, you've added a poly A tail on the other end, then you have spliced it and diced it, and now it's ready to go on to the ribosome to be a little protein. This is the step of translation. So the genetic information flows from mRNA to a protein through the process of translation. This is step 2, and it occurs out at a ribosome. Okay, so the molecular components of translation here, we're going to be dealing with a new type of RNA called transfer RNA. So the cell translates an mRNA message into a protein with the help of a transfer RNA or tRNA molecule. So tRNAs transfer amino acids. Makes sense. They're called transfer RNAs. What are they transferring? They're transferring amino acids to the growing polypeptide chain of a ribosome. This makes sense because without amino acids, you can't have a protein, right? So they are transferring amino acids. So translation is a complex process in terms of biochemistry and the mechanics, but we've got a couple different players here. So far, we've introduced tRNA and, of course, our mRNA that we talked about previously. Now we're in translation. We have opened up the DNA. We have copied the information onto mRNA. We have processed that mRNA, and now that mRNA has arrived at its final destination of a ribosome that we will translate the code that it is carrying. Here's a picture of what this looks like. Kind of complicated. When I draw this, because I'm probably going to draw it later, I draw it kind of like a Krabby Patty. That's what it looks like. You got a small subunit, you got a large subunit, and you got a space in between where you've got your mRNA that's being read, okay? So you can see we have our tRNAs. tRNAs are what? They are transfer RNAs. What are they transferring? They are transferring amino acids, okay? So you can see that they're dropping them off, and then all of these little amino acids are being added to our polypeptide chain because this is a protein. And what is the name of this? Protein synthesis. So we made exactly what we're after. That's a really crappy check mark. There we go. Okay, so let's look at transfer RNAs. This is the new RNA molecule. You're used to mRNA, our little single-stranded little guy that has uracil instead of thymine. Now, let's take a look at transfer RNA. So each tRNA can translate a particular mRNA codon. Remember, a codon codes for an amino acid. And a tRNA carries in that amino acid. So they play together very well here, right? Into a given amino acid. So the tRNA contains an amino acid at one end, and at the other end, it has a nucleotide triplet that can base pair with the codon. So let me simplify this. You have mRNA, and you have a codon, and a codon, and a codon. Let's say that you have like AUG, CCU, AGC. I made all this up, okay? Now it says that a tRNA contains an amino acid at one end, and at the other end has a nucleotide triplet that can base pair with a complementary codon. If this is a codon, what base pairs with this? A would pair with U because we're dealing with RNA. There's no T. U would pair with A and G would pair with C. This is called an anticodon, and it pairs up with the codon. And then you have that like amino acid It has an amino acid at one side and a complementary thing that's going to base pair with the codon called the anticodon. So this little guy is called tRNA. And that's how this looks all together. Okay, so a tRNA molecule consists of a single RNA strand that's about 80 nucleotides long. tRNA molecules can base pair with themselves. Flattened out, they kind of look like a little clover. They have like 4 little regions kind of, or 3 little regions and like a tail. In 3 dimensions, they look like an L shape. And the one end of the L contains the anticodon, and the other is going to contain our amino acid that it's bringing in because that's its job. So here's our pictures here. So kind of like a little clover, 2-dimensional, 3-dimensional looks like an L. Again, you have your anticodon and you have your amino acid attachment site. So you will have a little amino acid hanging off there. Okay, that's what they look like. So accurate translation requires 2 different steps. First, the correct match between a tRNA and an amino acid. So remember that the tRNAs are carrying amino acids. So the tRNA subunit has to be carrying the correct amino acid. And this is done by an enzyme called aminoacyl tRNA synthetase. So we're synthesizing, synthetase is going to synthesize a tRNA that has aminoacyl groups on it. So that's the amino acid. So second, a correct match between the tRNA that was just created thanks to this enzyme and the anticodon with the mRNA codon. So that's what I was just illustrating up here so beautifully for you. The anticodon and the codon pairing up correctly. That's the second step. There we are. So flexible pairing at the 3rd base of the codon is called the wobble position. It allows some tRNAs to bind to more than one codon. That's what I was talking about earlier. With like the last base doesn't really matter as much. This is where that comes into play. Okay, so step 1, amino acid and tRNA enter the active site of our aminoacyl synthetase. This is called tyrosyl tRNA synthetase because it's using tyrosine, which is an amino acid. So there's one for each different type of amino acid to help them correctly bind together, because it matters which anticodon is paired with which particular amino acid. Because if it didn't matter, then you'd have all kinds of wrong proteins being built. Okay, so it matters because the anticodon is going to pair up with the codon, and the codon says you need to bring in a specific amino acid, so they have to be complementary. So then you have the aminoacyl tRNA released once it is done. It uses ATP. Synthetase is going to catalyze covalent bonding. So then we have our completed little tRNA unit. Ribosomes. Ribosomes are going to facilitate specific coupling of tRNA anticodons to the mRNA codons during protein synthesis. The large and small ribosomal subunits make up a protein. So ribosomes are made of rRNA. So there's 3 different types of RNA. You have mRNA, which is our messenger. You have tRNA, which is our transfer. What does it transfer? It transfers an amino acid. And then you have rRNA, which is ribosomal RNA, which just makes up a ribosome with other proteins. So in bacterial and eukaryotic ribosomes, the large and small subunits join to form a ribosome only when they attach to an mRNA molecule. So like I said, the little halves of like our Krabby Patty buns, they don't attach until there's an mRNA molecule there. So this is what the overall process looks like. So like I said, I kind of like to draw them like little Krabby Patties like down here. There's 3 different sites because you're processing 3 different codons. There's 3 different steps. I'm dying over here. Is this the coronavirus? Oh my God. Okay. So you have the active, or the A site, the aminoacyl tRNA binding site. So this is where you're going to bind. You have an exit tunnel. You have an exit site. The P site is going to be our peptidyl tRNA binding site, which is where we're going to actually grow our chain out of that you can see here. And this is a much better drawing of what I just did. Okay, so you have our new amino acid being brought in right here. And then the growing polypeptide chain, then it will switch over, it'll move, and it will add its little amino acid onto the chain. So the next one will be a little square. And then it will exit. So it will go to the exit site and it will be released. So a ribosome has 3 different binding sites. The P site holds the tRNA and carries the growing polypeptide chain. Polypeptide, P site, makes sense. The A site holds the tRNA that's going to carry in the next amino acid. So this is like the entrance. And then the E site is the exit site. So they exist in this form, like EPA, like Environmental Protection Agency, but you start from here, APE. I like to think about this like attach, pass on the amino acid, exit. That's like the simplified version for me. So like A, attach, P, pass on amino acid, and E, exit. So this is the order that they exist in, but you go this direction. Okay, so building a polypeptide. So there's 3 different stages again: initiation, elongation, termination. You have heard these before. So all 3 stages require protein factors that aid in the translation process. It kind of moves you from one step into the next. So let's look at the ribosome association and initiation of translation. So the initiation stage of translation begins together with mRNA and tRNA with the first amino acid and the 2 ribosomal subunits. So the small ribosomal subunit is going to bind to the mRNA and a special initiator, tRNA. So this is a specific tRNA that carries in a specific amino acid that says, hey, we're going to start building a protein. Then the small subunit moves along the mRNA until it reaches a start codon called AUG. AUG is our first codon, always. On worksheets, will it be? No, because you're just making that up off the top of your head. Okay, but the actual first codon is always going to be AUG. So AUG is our first codon, and then we are going to have our anticodon that's going to bind there. And this anticodon attached to the tRNA will always bring in methionine. Methionine is going to be the first amino acid because that's what AUG dictates. AUG says the first amino acid is methionine, therefore it's methionine. So that will enter into our A site, slide over to the P site. Now we have our first amino acid. Our large subunit has come and attached, and everything is ready to begin. That is the initiation step. Okay, so the start codon is important because it establishes the reading frame for the mRNA, and the addition of the large ribosomal subunit is last and completes the formation of the translation initiation complex. Proteins called initiation factors bring all of these different components together to start the process of translation. So now we've initiated, we're ready to elongate. What are we elongating? The polypeptide chain, also called a protein because it's protein synthesis. So during elongation, amino acids are added one by one to the previous amino acid at the C terminus of the growing chain. So each addition involves a protein called an elongation factor, and it's going to occur in 3 different steps: the codon recognition, the peptide bond formation, and the translocation. So the translation proceeds along the mRNA in a 5' to 3' direction. We're going to look at this in some pictures in just a second. So here we go. So you have the recognition sites. So the anticodon will be binding to the codon in the A site. That's our recognition. And then you have the polypeptide formation. You have the bond forming between the amino acids. That's a polypeptide bond exists between 2 different amino acids. So you're kind of passing on. Remember, we're passing on the amino acid. So then we have our little empty guy here that's going to move over to the exit. And now we have our, our new tRNA that's then going to move to the P site. So that's what we see happening in step 3, translocation. We have transferred the location of our tRNA molecules here. So we are releasing one and we are growing our chain here. And now we are ready to receive the next one. Okay, so termination of translation. So once you have done this whole process 50 billion times, or however long that gene says, okay, there's this many codons to make this protein, sometimes they're really, really long, sometimes they're not. Just depends on the particular gene that you have transcribed and now translated. We have termination. So that's the stop. So termination occurs when a stop codon in the mRNA reaches the A site of the ribosome. So remember that there were 61 codons that make amino acids, and there were 3 codons that make a stop. When one of those stops comes into the ribosome, you stop. That makes sense. So the A site accepts a protein called a release factor. The release factor causes the addition of a water molecule instead of an amino acid, which is kind of like a, hey, we've reached the end. There are no more amino acids needed. So this reaction releases the polypeptide, and the translation assembly then comes apart because you are finished, and it will reassemble when there's a new mRNA. So this is the overall process here. Ribosome attaches to a stop codon on mRNA. The release factor promotes hydrolysis. You add a water molecule, which will cause the dissociation of our ribosomal subunits. So completing and targeting the functional protein. So often translation is not sufficient to make a functional protein. Sometimes you have many different polypeptides that make up a functional protein, which means that you would have to then attach all of them together, or sometimes there's extra processing that needs to happen. So polypeptide chains are modified after translation or targeted to a specific site in the cell where they're then going to either fully assemble or meet up with other things that they need to finalize themselves. Protein folding and post-translational modifications. So during synthesis, a polypeptide chain spontaneously coils and folds into a three-dimensional shape, which we know that we have like alpha helices and we have beta sheets are like some of our, simpler structures. And then we get into like our tertiary, quaternary, different structures that have multiple polypeptide chains all put together, just depending on the type of protein. Proteins may also require post-translational modification before actually doing their job. So targeting polypeptides to specific locations. Two populations of ribosomes are evident in our cells. You have free ribosomes that are just kind of floating around, chilling in the cytosol. And then you have bound ribosomes that are going to attach to the ER. That's what makes the rough ER. Okay, so free ribosomes mostly synthesize proteins that function in the cytoplasm or the cytosol. That makes sense because you make it where it's going to be needed. It doesn't have to travel very far. Bound ribosomes make proteins of the endomembrane system and proteins that are secreted from the cell. Because then in our bound ribosomes are in the ER, which is right next to the Golgi apparatus, which is going to help us accomplish all of these other targeting tasks. So protein synthesis always begins in the cytosol. Synthesis finishes in the cytosol unless the polypeptide signals the ribosome to attach to the ER. So that's how it becomes bound, right? That signal tells it to go become bound. Polypeptides destined for the ER or for secretion are marked with a signal peptide. So a signal recognition particle called an SRP binds to this signal peptide. The SRP brings the signal peptide and its ribosome to the ER. That's how it becomes bound. So this is how that overall process happens. You have the polypeptide synthesis beginning. The SRP binds to the signal peptide. The SRP binds to the receptor protein. The SRP detaches, and the polypeptide synthesis resumes. You have the signal cleaving enzyme cuts off the signal peptide, and you have the completed polypeptide. It's going to fold into its final conformation. So making multiple polypeptides in bacteria and eukaryotes. In bacteria and eukaryotes, multiple ribosomes translate an mRNA at the same time. Once a ribosome is far enough past the start codon, another ribosome can actually attach to the mRNA and continue to transcribe itself. So strings of ribosomes are called polyribosomes or polysomes and can be seen with an electron microscope. They kind of look like little beads on a string. So these are a whole bunch of different ribosomes that are transcribing the same RNA. It's mRNA. It's just a really, really long one. Again, this is in a bacterial cell. Bacteria and eukaryotes can also transcribe multiple mRNAs from the same gene. So in bacteria, the transcription translation can take place simultaneously. And in eukaryotes, the nuclear envelope is physically separating these two processes. And that's what we talked about earlier. So here's the picture you can see here of our RNA polymerases, DNA, because this is our DNA, and then immediately you have your mRNA coming off of that. You know that this is inside of a bacterial cell where you have these things happening at the same time. So this is kind of like the overall process in eukaryotes. You have transcription up at the top, which is when we're going to take that DNA, open it up, make our RNA transcript. We're then going to go through RNA processing. So we're going to take that pre-mRNA, cut out the introns, sew back together the exons, make sure you have a poly A tail and a 5' cap. And then that finalized mRNA can go into the cytoplasm, the cytosol, and it's going to meet up with the ribosome. The ribosome is then going to translate the codons. The codons exist on our mRNA. AUG is always our first codon that brings in signals to bring in methionine. Our tRNAs are going to be bringing in all of our different amino acids to grow our polypeptide chain. When you reach the termination sequence, then you are finished making your polypeptide. And this is the overall process of protein synthesis. So some stuff can go wrong in all of this processing. So mutations of one or a few nucleotides can affect protein structure and function. And a lot of times, the mutations can actually render them like useless. So then they just like get broken down, recycled, and then the pieces can go back to make different proteins. So mutations are changes in the genetic material of a cell or a virus. So point mutations are chemical changes in just one or just a few nucleotide pairs of a gene. So point mutations are very small. So the change of a single nucleotide in a DNA template strand can cause the production of an abnormal protein. So if just one letter, one base is wrong or incorrectly paired or whatever, it's going to cause a point mutation, which can create an abnormal or non-functional protein. So you have wild type hemoglobin and sickle cell hemoglobin. So sickle cell disease is when you have red blood cells that are sickled in shape and they don't carry oxygen efficiently because of their altered shape. And it's actually caused by a point mutation in one single position. So instead of making glutamine, you're going to be making valine. And that's one amino acid that is different. And now you have a mutant hemoglobin protein. So point mutations within a gene can be divided into two general categories: a nucleotide pair substitution, or one or more nucleotide pair insertions or deletions. So here are a couple different examples. It just kind of goes through it, tells you the names of all of them. You have a substitution, which you can see happening here. You have an insertion deletion. You have an extra A. So all of these different mutations, we have silent, missense, nonsense, frame shift with one nucleotide deletion or three, right? So we'll go through and look at all the different types of mutations that are outlined here for you. Okay, so substitutions. We have a nucleotide pair substitution, replaces one nucleotide with its partner and its partner with another pair of nucleotides. So think about like a substitution during the day, like if you have a class period or something where you have a sub first period. Does it affect the rest of your day? No, it just affects that one particular class period. And that's how these nucleotide pair substitutions work. They affect one amino acid typically, not more than one. So it's just one thing that's actually being affected here. So we have silent mutations that have no effect on the amino acid produced by a codon because of redundancy in the genetic code. So remember CCC, CCU, CCA, CCG, they all coded for proline. So if you have a mutation that occurred here, you would never know because you would still make exactly the same amino acid called proline, which means that your polypeptide is not going to be messed up because it still contains the right pieces. So here's an example of our silent mutation that you still make the correct amino acid at the end. So there is no issue here. And that's happening here. So then we have missense mutations. They still code for an amino acid, but it's just the wrong amino acid. So the substitution mutations are usually missense mutations. So if it's not in that wobble position, that third position, if it's one of the first two in the codon, typically it's going to be a missense mutation. And that's the example of our sickle cell anemia. Then you also have something called a nonsense mutation, which is a change in amino acid codon and a stop codon. So instead of coding for an amino acid, now you code for a stop. And they always lead to a non-functional protein because basically, instead of adding in a valine or a serine or a glycine, you're now going to say stop and you haven't reached the end. So it's not going to be functioning because you haven't finished yet. So here's an example of a missense. So you have the T instead of a C. So you are going to have your glycine is going to become a serine, right? So now we are creating the wrong amino acid. And then your nonsense is where you are going to have a stop codon put in instead of the correct amino acid. And all of this, like nothing else happens. A lot of kids get confused about that. Because you've reached a stop, then that causes the water molecule to attach to our polypeptide chain. In this case, it's just methionine attached to a water molecule, which then causes the ribosome to dissociate. So it can't keep going because it's not together anymore. Insertions and deletions. Insertions and deletions are additions or losses of nucleotide pairs in a gene. These mutations have a disastrous effect on the resulting protein because substitutions are only changing like one specific place. There's one thing that's being affected, whereas the insertion and deletion changes the total number of nucleotides, which affects everything downstream. So anything that comes after the mutation is affected. So the insertion or deletion of nucleotides may alter the reading frame of the genetic message, producing a frame shift. So the two different types of mutations are point and frame shift. Point are a lot less disastrous, while frame shift are very bad. So let me give you an example. These are all 3-letter words that make a sentence. The fat cat ran. Okay. This works for codons because you read 3, like codon 1, codon 2, codon 3, codon 4. You read 3 bases at a time. Let's say that I have an insertion. Let's say that I insert an R right here. THE is fine. Nothing happened. But then I go 1, 2, 3, FAR as my second codon. And then TCA. And then TRA. And then I have an extra N that doesn't really make anything. Is this the same thing now? No, it's completely different because I added one base. When you change the total number of nucleotides, you've screwed up everything, right? Because now you've changed the reading frame, which is called a frame shift mutation. So this is an example of a frame shift mutation causing an immediate nonsense. Because you added in an extra A, it codes for an extra U, which creates a stop immediately, which causes the dissociation of the ribosome. And then the next one, you have extensive missense. So basically, when you add in, this one is a deletion. You've deleted one of your pairs, which means now the reading frame has shifted because you've deleted. So instead of my example where I added one, it's like they took something away. Well, you're still going to shift everything. You're going to have 2 extra letters at the end instead of 1 if you have a deletion. So now this is an extensive missense. So all of the amino acids that are made from here on out are going to be the incorrect ones. This is a no frame shift, but 1 amino acid is going to be missing. So if you actually have 3 that are deleted, you're missing an entire amino acid here, and that can happen as well. Which means that at the end, you're still not going to make the correct polypeptide. It probably will not function. So then we have something called mutagens. So mutagens are spontaneous mutations can occur in DNA replication, combination, and repair. They can happen kind of at any time. Mutagens are physical or chemical agents that cause mutations. And we know that like cancer-causing chemicals are called carcinogens. These are definitely mutagenic, which means that they can cause mutations. Researchers have developed methods to test the mutagenic activity of different chemicals. And it's really sad that we are still selling these chemicals on the market, like in tobacco products and cigarettes. They are mutagenic. They are carcinogenic. They cause cancer because they are causing mutations in the DNA. And mutations in the DNA, as we just saw with our frame shift and point mutations, change your proteins. That can change your physical appearance. It can change any of the enzymatic activity in your body and prevent it from functioning. Any of the, the proteins that actually make up different parts of your cells can be damaged because their genes are no longer correct because they are mutated. So there's a whole lot of things that can happen because of mutations. Most of them you learn in like evolution are neutral, but if you have a mutation in your DNA that is a disadvantage because it's preventing the correct protein or polypeptide from being created, now you have this like disastrous chain of events that's going to go on each time that cell is replicated. So what's a gene? Revisiting our question from the very beginning. The definition of a gene has evolved over time. It's changed a whole lot. We've considered a gene as a discrete unit of inheritance, dealing with more like Mendel, the region of specific nucleotide sequence in a chromosome, more like Morgan, and the DNA sequence that codes for a specific polypeptide chain with Beadle and Tatum. So a gene can be defined as a region of DNA that can be expressed to produce a final product, functional product, called a protein, either a polypeptide or an RNA molecule, okay, just depending on the type of organism and which gene it is specifically. So basically, a gene is a section of DNA that can result in a product, a functional product, that's typically a polypeptide or RNA-containing molecule. So that's going to be it for protein synthesis. The next chapter, I believe, goes over gene expression and how to upregulate or downregulate which sections of DNA can actually be transcribed or translated. So it's going to build directly off of this. I know that this was a long one and it's a little bit complicated, and I know that I'm coughing and I'm sorry I'm sick, but I'm trying to get these out for you guys to help you out with your upcoming exams. So thanks for sticking this out with me, and I hope you have a great day today.